function in different, what will be the activities of our cells. All these information are encoded in the genes. But the genes are basically inactive. Why I am saying inactive? Because unless genes become active, the cells, they cannot function. They are basically sleeping. The genes are sleeping, and when they become active, their function begins. By activation of genes means that this is a two-phase process. One is called the transcription. By transcription, a DNA or a gene, specifically a gene, is transcribed into an RNA. So RNA is similar to DNA, ribonucleic acid, but it is AUGC. That instead of thiamine, we have uracil, and it is a single-stranded. So from DNA, it creates messenger RNA, mRNA, and from RNA through translation, another process, another biological process, through translation, some proteins are created. Proteins are again some strings, but they are not ATGC string or AUGC string, rather they are strings of amino acids. So anyway, the proteins, the thing is that the protein is very important because its proteins are the active parts in the cells. All the functions of the cells, all the activities of the cells are actually performed by these proteins. So until and unless genes are converted into proteins, although genes have the information what function is performed, but until and unless gene is converted into protein, that function cannot be performed. So in computer science point of view, we can say like that we know the difference between a program and a process. A program is a passive entity. It is sitting in your hard disk. It is doing nothing, but it has all the information, how it will perform. But when it starts executing, it becomes a process. Then it starts functioning. So similarly, gene is the program and the protein is the process where it is performed. So what happens, the one interesting thing is that in our cells, all the cells in our body has the same set of genes, exactly same set of genes. Then the question is, why the different types of cells perform differently? For example, the cells in our lung, their function, the cells in our hair, their functions are completely different. But they have the same information, same set of genes, they are in same program. Then why their functions are different? This is because not all the genes are active in all the cells. The set of genes, those are active in our lung cells. This set is different from the set of genes which are active in our hair cells. Again, that is different which are active in our skin cells. Right? So active means what? That the genes which are creating proteins. So these sets are different from one kind of tissue to another kind of tissue. So therefore, these phenomena, so when a gene starts to create proteins, we say that gene is expressed. So this phenomena is known as gene expression. And different genes, amount of expression of different genes are different in different cells, different kinds of cells. And how it is measured? It is measured that how much RNA a gene is producing. The amount of RNA the gene is producing is called the expression level of that gene. So from this phenomena, the biologists have produced some tools which can produce a data like this, which is known as microarray gene expression data. Microarray is nothing but a matrix-like structure. We are interested only with the data. I am not interested with the biology, how that is produced. Okay? We are interested with the data. So it is nothing but a matrix where the rows represent the different tissue samples. Tissue samples means that, for example, you want to use this data for cancer classification. So what they do, that they collect some tissue from some cancer patients. For example, if you are dealing with the lung cancer, so with the patients who are already detected to have lung cancer, they have malignancy already detected, they collect samples from those patients. And from each of these samples, they compute or they find that what is the expression levels of the genes in these samples. Okay? So suppose these are the malignant samples. Sample 1, sample 2, sample 4, 5, 6 are malignant samples. I have shown here only 3, but there are several actually, 50, 60 samples. Right? May not be the same patient, from different lung cancer patients. Similarly, the patients which are known to have non-malignancy, that is, which are detected to that their tumor is non-malignant. Those patients, from those patients also some samples are collected, and the expression levels of the genes in those samples, the same set of genes in those samples are also computed. So in the rows we have the samples, maybe two types of samples, normal samples and malignant samples, and in the columns we have the genes. And the value in the cell ij in this matrix represents the expression level of the jth gene in the ith sample. Jth gene in the, because in the columns we have the genes, but you can transpose this out. So here we have shown the genes in the columns, so this value represents the expression level of gene 2 in sample 3. Okay. So this is what we call microarray gene expression data. Now what these colors represents, so this is, we represent it by some colors to ease the visualization, for the purpose of visualization. We actually give some shades to these values. Higher the value, means the higher expression value, we give some shades of red color. And the lower expression value are given some shades of green color. More greenish means more lower is the expression. The reddish means the higher is the expression. And blackish means that neutral expression, not very low, not very high. So here given, so your problem is that, let us first define the problem. So you are having a microarray gene expression data with two kinds of tissue samples, malignant and normal, and you have several, maybe 1,000 genes whose expression levels have been computed on these two kinds of samples. Now your job is to find those genes which have different expression pattern in these two kinds of samples. For example, some gene may have very low expression in normal samples, but has high expression in the cancer samples. So these genes are called as oncogenes. Oncogenes means onco from oncology. So oncogenes means that the genes which don't express normally in normal tissue samples, but when they start expressing, that means some mutation has happened into that gene. So they have started expression, they have started to create proteins. So that becomes malignant. That means that leads to malignancy. So that's why these genes are important as a marker of malignancy. So if we are given an unknown sample, you suppose don't know whether that is malignant or normal sample, what you will do, that you will try to find what is the expression value of that gene. If that is high, then there is high chance that sample is malignant. So it helps in diagnosis.
Correlation.
Yeah, It helps in diagnosis and also maybe in future some drug design. If you want to just negate that process, that means the genes have somehow has been turned on. If you can somehow turn it off again, that may help in preventing or dealing with the cancer. The other kinds of genes does the opposite. It's also possible. That means in some genes which are highly active in the normal samples, but in cancerous samples they don't express. That's the opposite. So these genes are known as tumor suppressor genes. The genes, when they are active, they suppress the tumor. When they become inactive, the tumor goes. So these are also markers. So these identifying these marker genes is very important to us because they help in diagnosis and also in drug design. Because the people can design drugs targeting those genes. So these genes can be taken as the targets, drug targets. Well, so how we can pose it in a computer science problem? Like, so gene markers are those the genes of microarray acid that correctly classify the normal and malignant tissue samples. Just seeing the expression pattern of the gene, you can classify that whether the corresponding sample is malignant or normal. So gene markers can be very good indicator of cancer, helpful in diagnosis. So gene marker identification problem from a microarray gene expression data that can be posed as a feature selection. Why? Because here genes will be treated as features and we have to select those features which maximizes the classification accuracy of your two kinds of samples. It may be two kinds of samples or it may be multi-class data also is possible. That multiple types of cancer. Which type of cancer you have got, so that also can be classified. So identifying the marker genes can be posed as a feature selection problem. So the general flow will be like this, that original data set with all the genes. We somehow select a subset of genes. We evaluate the selected subset in terms of some performance measure. That how these selected subset of genes are performing in terms of classification, and that will go on until we are satisfied we are getting the sufficient classification approach. So this part can be implemented using multi-objective technology because you can have several objectives. One objective is to maximize the classification accuracy. You want to select those subset of genes which maximizes the classification accuracy. Additionally, you want to select that subset of genes with the minimum set of genes that classifies the data very well. So minimization of number of selected genes, that can be another objective.
And the feature selection.
Feature selection. So two objectives we have already defined: the classification accuracy, maximization, number of selected features, minimization. There may be other objectives like precision, recall, sensitivity, specificity. There are several. Any classification performance metric can be treated as an objective function. And you want to optimize all these objective functions simultaneously. And you have to identify the set of genes that optimizes all these objective functions simultaneously. Right? So that's why we can say that it is a multi-objective optimization problem. Now to evaluate the selected subset, that means suppose, so what here I am doing that, what is a possible solution? Possible solution is a subset of genes. So we encode a possible set of genes in a chromosome. And to evaluate that set of genes, how good that set of genes in classifying, for evaluating this we apply some classifier. We can apply neural network classifier or any other classifier, support vector machine or any other classifier, right, to compute the classification accuracy through some cross-validation technique. I am not going into detail, but it can be done. So evaluation can be done by some classifier. So that I said that. So how the encoding will be now? So my chromosome here, chromosome is in terms of genetic algorithm, not the original chromosomes. So chromosomes or strings will be used for encoding only a subset of genes. From the total set of genes, the chromosome should be able to encode a subset of genes. So if you have suppose total 10 number of genes, we design a binary chromosome. Binary chromosome of length 10. So you see that here you don't have to input L, because it will depend on the problem that how many genes you are doing. So you have a binary string of 1, 0, and the string length will be 10 as you are dealing with 10 genes. Now what these binary values will represent? If the ith bit is 1, that means we say that ith gene is selected. If the ith bit is 0, that means that gene is not selected. So this is how a binary string will define a subset of genes from the complete set. Only the ones will be, that those indices, those genes will be selected in that subset. Okay. So for example, this is one chromosome, so corresponding subset of genes here is. So initial population, again we can create random. So once we have created that subset of genes, so this blue ones are the subsets, that those genes which are selected. So we project the data on this subset of genes with all the samples, but only these genes. Then what we do? We apply some cross validation. I am not going into great detail because I don't have any time for that. So we apply some cross validation technique with a classifier, wrapper classifier, to find the classification accuracy, or other classification metric also you can find, sensitivity, specificity, precision, recall, etc. So that can be one of the objective functions.
Sir, how do you perform the training and classification in
Yes,
this
that cross
particular-
validation is done. That's exactly.
Okay.
Cross validation, there are several cross validation. Here you can look the cross validation. It's leave one out cross validation. Suppose all the genes, you know that. You can just
Based on the worst are eliminated.
Yeah.
Yeah.
So basically, what we are doing, that suppose we have total 50 samples. One sample is left out, and with the remaining samples you train the train, and the left out sample is tested, so predicted. So that is repeated for each of the samples, and then we find out what is the overall accuracy. Right? And another fitness function, as I said, that it can be a number of features selected. So we want to minimize this, we want to maximize this. Okay? So that is how we can proceed as a multi-objective optimization problem. So selection is again normally the crowded binary tournament selection that is used in SJ2. Crossover, we have used the single point crossover. You can use the other type of crossovers also. And mutations include the mutation. These are some example parameters mainly found by some experiments. So here is an issue that, as in the multi-objective optimization, as we have seen that from the final generation, we will get a non-dominant set of solutions, not a single solution. We will get a non-dominant set of solutions. So again, the question comes, which will be my final solution? So one thing can be done, that as if we give more importance to the classification accuracy, then we choose the solution with the highest classification. Or what you can do, that you can combine all these solutions and find out the common set from that, which genes are common in all these non-dominant solutions. That can be done. So that can be designed, that will depend on the problem. Some results are here. So here, this is the complete. So this is a colon tumor data set. So these are the publicly available data sets. So this is the complete data sets with several genes. So these are the selected set of genes that. So you cannot expect that all the genes will be uniformly red or green, because microarray data sets have a lot of noises, because they have been generated from biological experiments. So a lot of noises will be there. So you cannot expect that 100% accuracy. But so you can easily see that these selected genes, these two genes, these are having no expression or low expression value in the tumor samples, and in the normal samples they are highly active. So basically they are tumor suppressor genes, because when in the normal samples they are highly active, in the tumor samples they are almost not active. On the other hand, these set of genes, they are active in the tumor samples but not active in the normal samples. So these are oncogenes. These are one example. So this way we can apply genetic algorithm on feature selection. This is another example for leukemia dataset. You can see that again. So here the two types of leukemia. Here the two classes are two types of leukemia, ALL and AML. So here are some useful links. You can see that is the fungal link. In ECI machine learning repository, so this has lots of data sets, real life data sets there. Not only the biological data set, but general real life data sets from the finance, facilities on business, from different scientific points of view. So lot of data is there. You can get there. And these are some links of microarray data sets from where we have collected the data. Public data sets are public. Get that. So here are some references. Most of them are our papers. You can go to my website and find the papers from that. So here is one book that we have written that has mainly come out of my PhD thesis. It was published last year. Here we applied multi-entity genetic algorithms for data clustering problems and with some applications on remote sensing and bioinformatics.
So can you go back to the literature?
The first paper is the NSG paper, the Poland paper, 2000 evolution. The rest of them are our papers.
Most of them are relation.
Yeah, I never done that. Yes, Bandhavade is currently director of
ISI.
ISI code.
ISI.
Sir, your book will cover every aspect of multi.
Yeah, there is. Actually, the book is very costly, because this is a foreign publisher, so it cost around 4,000 something. But what it covers what that it has a general introduction to genetic algorithms and also the multi-objective genetic algorithms and the applications of remote sensing and bioinformatics. That part we performed mainly in the PhD, my PhD. And what you can do to have a general idea of multi-objective evolution algorithm, we have written a very detailed survey of multi-objective evolution algorithms on cluster recently. That was published in the same monthly survey. So, if you want, I can do it. You can get it from my website actually. And these two are also very. So this one is also recently published another survey report of ours that this multi-objective evolution algorithm for data mining problems, all kinds of data mining problems, not only clustering. It was feature selection, clustering, classification, association, rule mining. Everywhere the people have applied data multi-objective optimization, multi-objective genetic algorithms. So we did a detailed survey of that. Actually, the paper has two parts. This is part 1, another part 2 is there. In the same volume of the journal, it was published. that also is available from my website. I will just give the link of my website. So if you have any questions. So this is the link of my website and this is my email. If you are interested, you can mail me. And you will find most of my papers are uploaded in this website. You can get it. At least the journal papers are there, not all the conference papers are there, but journal papers are uploaded. So any questions, please?
Sir, apart from MATLAB, you have any other programming or particular type of software where we can find this type of programming, where we can use our application?
There is R also. So R. So actually the MATLAB is, you know that it is not open source. It is
No,
costly software.
it's not. Yes.
So
That's why.
we usually use the pirated version. So
Yeah.
it is open source.
Absolutely.
But there is an alternative of MATLAB that is open source. That is called R. So you can it's called R Lab, and that is several statistical tools packages are already given there. You can use that.
Even the NSJ2,
Yeah, you will find the package for genetic algorithm also.
the NSJ2.
I am not sure NSJP is whether is there, but actually, if you ask me that we prefer to write our codes, that I am
No,
not very
definitely.
interested to find out the packages where these are already. Because it is, first of all, it is a fun to write code myself, and also it is better to write your own code because it is easier to modify later on.
Actually, his background is basic physics.
My background is.
That is why he is here.
Okay. So in that case, MATLAB is more.
I normally work on multi-phase flow system, so
Okay.
that's a different. The application of neural networks and this is on that particular field, so it's completely different from this. But since we use it as an application, that's why I'm very interested to know if there is anything software or anything like that which is available.
I'm not sure.
Like
but,
in neural network, there is a software called Weka, very
yeah,
new.
Weka is there.
From-
Weka, not only in neural
Okay.
network, you will find most of the data mining algorithms are implemented in Weka.
You can
lot
find
of
these
that
things
class-
also.
uh,
Genetic
No,
algorithm,
genetic algorithm is not there. That is for data mining.
data mining
So
purpose.
several clustering algorithms are there, classification algorithms, code vector machine neural, all of them. Some rule mining algorithms are there. So Weka is basically for data mining, not for genetic algorithm.
Okay.
Right?
For genetic algorithms,
For genetic algorithm, I don't know really on that because I have not used any packages.
Okay.
That, maybe I can search it. That you can just send me a mail. I will give you something.
What kind of researches are happening related to energy?
You see, the NSJ2 is already 14 years old, but still the people are heavily using it. As I said, that it is the most cited paper in Mukhopadhyay literature. But lot of modifications also have come out of NSJ. Because one problem is that if you have 3, 4, 5 objectives, that's fine. But if you have 100 number of objectives, then these NSJ2 or these kind of algorithms will not be able to deal with it. Because the problem is that if you have 100 objective functions, what will happen? So almost all the solutions in the population will be non-dominating, in the same rank. Right? So it will not work. So these are called the many objective optimization, not multi-objective, but the many objective optimization. And there some researches are going on, like how can you reduce the objective space by finding the redundant objectives or unimportant objectives, like that, reduce it a smaller space and then. And also there is some interactive genetic algorithms are coming. Interactive means is whether you can help the genetic algorithm, because sometimes the people have more expertise. But as there are a lot of solutions, it is difficult to apply their expertise to find better solutions. But what the genetic algorithm is doing, they are minimizing the total number of solutions, that out of 1,000 solutions, suppose it has come out with the one, after certain number of generations it has come out of the 50 solutions. Then the people can identify, okay,